session on animal models, where we are trying to understand how they interact with plant specialized metabolites, aka phytochemicals. So in this case, in my research, I try to understand TRPV1-independent and sex-specific behavioral traits and signatures for capsaicin in the fruit fly. Okay, so we know that capsaicin is the main pungent compound associated with the spiciness sensation in mammals. And the basic mechanism of spiciness is through the activation of TRPV1 in nociceptive neurons. And we also know that this spiciness sensation is likely exclusive to mammals. And this is a way of plants to develop a coping mechanism to block the seed digestion that mammals don't have. So, as I mentioned, the underlying mechanism of spiciness is TRPV1, and in this particular channel we have a capsaicin pocket. It acts as an antagonist, and then there are 4 key amino acids associated with this sensation: serine, tyrosine, threonine, as well as glutamine. And we know that point mutations that change the translation of these amino acids cause the capsaicin sensation loss, or at least the less sensitivity. So in this case, we are using flies because they are a good trackable model that we can use to understand how genes impact behavior and physiology. So in this case, one of the objectives that we had is to understand the evolutionary relation of trip channels. And we found that there is a clade specific for invertebrates where the fly homologs are present, whereas we see that for other vertebrate models, this clade is divergent. And we also don't observe any conservation of these key amino acids. And then when we visited the literature regarding capsaicin and Drosophila, we found contradictory and confusing results. Therefore, we proposed 2 frameworks to understand how the flies interact with this compound. One where we have sensation through the activation of low affinity TRPVs, and another one where capsaicin is able to stimulate gustatory neurons. Whereas in the case of no sensation, we think that the effects of capsaicin in the fly is through a high capacity to metabolize this compound and the indirect activation of TRPs by reactive oxygen species, since we know that these species can activate these channels. And of course, these responses are dose dependent. So from this, we can conclude that the classification of model organisms can be divided into 3. Those that are capsaicin sensitive, capsaicin insensitive but possess TRPV1, and those that do not have TRPV1, such as the fly. And then, because of that, we cannot say that the flies will behave as humans, but we don't know exactly how they interact with the compound. So in my research, I'm trying to investigate these TRPV1-independent responses, as well as the underlying molecular and chemosensory mechanisms. And I propose 3 objectives. One is to dissect the TRPV1-dependent and independent responses. The second objective that I have is to understand through a transcriptomic data set the underlying molecular mechanisms at a single cell resolution. And then I also wanted to identify the neuronal populations as well as gene regulatory networks underlying these behaviors. So I divided my presentation into 2 sections. I try to go as quick as possible in this section, where we basically compare TRPV1 dependent and independent responses through a humanized model. So we use these genetic lines. Basically, we use 2 wild type strains, and then we use the US Gulf system which enable us to basically express a particular gene into a particular cell type or tissue. So in this case, we are expressing TRPV1, the humanized version, in multidendritic neurons. Those are nociceptor neurons of the fly. That way we can say we phenocopy the effects of TRPV1 in the fly. And then just, this is the pattern that we use for the TRPV1 expression. This is the middle brain, and then the hindgut of the fly. And for behavior, we first wanted to understand the capsaicin taste through proboscis extension reflex. And then for binary choice, we use the two-choice experiment in order to understand dietary decisions. And then we also measure food intake through the capillary feeder assay. Okay, so first, we observe that the flies have a proboscis extinction response as you increase the capsaicin concentration. And this is not strain dependent, since we discovered the same for the two wild-type strains. And we also observed that the capsaicin addition to sugar is able to increase the reflex response of this palatable component, which is not the case of the humanized version of the fly, where these flies basically abolish the capsaicin diet, even in the presence of sugar. And then for the binary choice, we see that the distribution of the preference is increased as you increase the capsaicin concentration. Whereas for the TRPV1, we see a complete opposite effect when we supplemented the flies with capsaicin. So from now on, what you're gonna see is just experiments with 2 millimolar of capsaicin. So we fixed this concentration since it gave us a good phenotype. We also consider this concentration as naturally relevant because it is found in chili peppers. And then it was technically possible because if you increase the concentration of capsaicin, you start to see some precipitation on your diet. Okay, so what you are observing here is that the flies, the wild-type flies, especially females, increase their food intake at the 24 hours mark. This is not the case for males, and the opposite trend is shown for TRPV1 positive flies. So what's interesting here is that after the 24 hours, flies undergo feeding habituation. That means they transition from a hedonic feeding to a homeostatic feeding, indicating that there might be an underlying mechanism that is regulating feeding satiety. So from these results, we can conclude that the wild-type flies have an attraction to the capsaicin diet, whereas the humanized flies are repelled. And then after the increase of feeding, they undergo feeding habituation, which is independent of TRPV1. So we know that feeding behavior is the integration between sensory cues as well as metabolic signals. And we also know that activity patterns integrate these signals to optimize food search and nutrient intake. So in order to understand if there is any metabolic influence of capsaicin in flies, we measure 4 parameters: body weight, glucose, protein, and triglycerides. And then we also measure activity by using infrared beams. And then we analyze the sleeping traits with the assumption that the sleeping in flies is considered every 5 minutes bout without activity. Okay, so in summary, we didn't find any effect on the body weight for the wild-type flies, where this is true for TRPV1. And in the protein levels, we didn't find anything significant. So indicating that body weight reduction is TRPV1 dependent. Same case for triglycerides. We only observed significant changes for the humanized fly for both females and males, indicating that the fat loss at the lower glucose levels are TRPV1 dependent. So now moving to the activity pattern, we only observe disruption of total sleep in the humanized flies, whereas for water flies we don't find anything significant. However, when we go to the activity patterns, we see that the water flies do have a reduction of activity, but only occurs at night. And we also see that the female flies have disrupted sleep. So what's really intriguing here is that the reduction of activity only happens at the evening peak, a phase where the flies also control their feeding rhythm, which I'm going to address later, but this is going to be important for our transcriptome data set. So from this part, we can conclude that there is no influence of capsaicin in the metabolic profile of the flies, and there is an inhibition of activity. So then we ask, because we know that the gut can drive also dietary decisions, whether the gut has any influence on these phenotypes. And for that, we did raw detection through DHE staining in order to understand if the chronic exposure of flies activates any neuronal response in the gut, as well as if the metabolism of flies is also provoking any induction of ROS. And then I want to thank other lab members because we have a side project where we are trying to understand how phytochemicals influence the microbiome as well as the gut transcriptome. So here I use some of their data in order to understand if there is any neuronal signaling in the gut. So in summary, we only found that the oxidative stress is TRPV1 dependent, particularly in the midgut of the flies when you supplement them with capsaicin. Therefore, there is no ROS activation in water flies when you supplement them with capsaicin. And then for the good RNA-seq, this is just water flies. We see influence on the transcriptome when you supplement them with capsaicin. And we found some shared genes, though they are mostly sexually dimorphic. However, when we look at the gene list, we only found metabolic signatures, but we didn't find any neuroactive signaling or any neuropeptide expression, indicating that there is no activation of neurons in the gut, at least for our data set. Of course, we cannot rule out that possibility, but we don't have any evidence that it shows gut has any role on these dietary decisions. So we can say that the feeding behavior and the activity show an opposite effect on wild type and TRPV1 positive flies, whereas the metabolic impact is strictly TRPV1 dependent. And I want to be clear here that at the tested concentration, because this can change depending on how much you supplement them with capsaicin, these effects are unlikely the result of good mediated sensing or metabolic needs. So now, for the second part of my research, we constructed a transcriptomic data set at a single cell resolution where we're trying to understand the capsaicin signatures that are driving these behaviors. And we also identified some neurons driving the capsaicin response. Because we know that the brain controls behavior through sensory inputs as well as metabolic signals that are defined in neuronal circuits. So here, two important factors that we consider is the fact that there's a delay between the mRNA and the proteins expressed that regulates the circadian rhythm. And we also know that the feeding behavior is controlled by this clock. Another curiosity of the flies is that they are not diurnal nor nocturnal. They are rather crepuscular animals because they only have activity in the morning and in the evening. So what we try to do here is to capture the transcriptome response in this time frame of the day where we observe the phenotype. So we pull brains and then we send for sequencing. They basically perform tissue lysis and then they purify nuclei and then they detected the viable nuclei and then they perform the sequencing of each nuclei through DNA genomics, which basically captures and constructs the library for each droplet. And then we perform the analysis, some standard methods and also some emerging methods specific for flies. Okay, so here what you can see is the data set for each sample. And what I want to express here is that we collected very similar brain representation, which is good, so that way we can compare our different conditions. And then we estimated the cell proportions here, and then we clustered them into different cell types. However, what's important here is we know that these are probably different cell types based on the clustering pattern, though we don't know which cell is, for example, this cluster or this other cluster. So in order to do that, we basically performed DEX analysis between all the clusters, and then we collected the genes that are mostly expressed in each cluster in order to annotate these cell types, consulting the fly cell atlas as well as the fly brain atlas. And then we ended up with this picture to depict the data set. I actually have to tell you that I spent around a month or a month and a half just for this figure. For now, we can say different cell types so that way we can understand the different signatures in our data set. And then we perform a screen of differential expressed genes. So here I want you to focus on three things. So the first is that we have shared genes that are in yellow. Then we have female-specific and male-specific genes that are in green and red, respectively. What I want to highlight here is that we found DSK, or Drosophila, which is a neuropeptide that is shared for females and males. Then we also identified clock as a shared signature, which is the K modulator of activity patterns. So when we go to the literature, we found that DSK release is associated through food satiety, and through DSK, sensory neurons are desensitized, which suggests to us that this neuropeptide might act as a way of the flies to induce food satiety. So we wanted to see what are the cells expressing this gene, and we can do so in our data set. So we basically dig down in our data, and we found that this gene is indeed expressed in peptide neurons, and there is a clear differentiation between females and males, and also between males with capsaicin and females with capsaicin diet. And we wanted to see if this gene is expressed in insulin-producing cells or not. And we found that it is a cluster associated, but it's not co-expressed with DSK, indicating that peptide neurons are indeed related to DSK release, but not those expressing insulin-like peptides. So with this, we can suggest that DSK likely modulates feeding habituation in flies, expressing peptide neurons. So now, what might be controlling activity? When we consulted our pseudobook responses, that means the comparison between data sets for each specific cell type, we found that glial cells are mostly responding to the capsaicin diet in both females and males. And turns out that in this list of DEX, we found clock, as I mentioned, the master regulator of circadian rhythmicity. And we also find neuropeptides that are known to regulate the evening activity peak and are normally co-expressed with clock. So we wanted to know if correlation is also, because correlation is not sufficient, we need to understand causality. So we first asked whether this gene is present in the glial cells, and those are the glial cells that mostly express clock in the capsaicin diet. Mostly cortex glial, enchanted glial, and a cluster of unidentified, but that we know is glial cells. So we perform a scenic, that is a machine learning approach to understand correlation between gene expression. And then to understand causality, we mapped the motifs of the genes that are correlated to clock to see if there is any interaction of clock into their upstream binding domains of these genes. So what's important here is that the clock regulon, we're going to use the regulon as the genes that are regulated under the same transcription factor, are mostly expressed in this region of glial cells, as you can see here. And we found that this clock regulon is indeed mostly expressed under capsaicin diet as comparatively to controls, indicating that there's a clock-dependent mechanism that is reprogramming the transcriptional landscape of the flies under capsaicin diet, which influences neuropeptide expression. So now we are going to the sex-specific traits. We didn't find any male dopaminergic decks, but in females we see a lot of decks related to neuroactivity and the calcium cascade that is normally produced when neurons are active. So we were wondering if there is a lack of dopaminergic valuation in male flies, but is present in female flies in their learning and memory circuit, which basically defines mating, courtship, and feeding behaviors. So indeed, we see a strong response of dopaminergic neurons when we see the cell-to-cell communication analysis, whereas for males, we see mostly a pituitary bias, which is related to locomotion and conversate, as represented as a DSK signaling, indicating that whereas for males there is a dopaminergic bias, whereas for females there is a dopaminergic bias, for males a peptide bias.
Explain marking rates.
I'll address that later, which might impact behavior, of course. So now, transcriptions are okay, but of course, sometimes we have a stochastic expression. So we wanted to confirm our expression analysis with bulk RNA-seq. So for that, we use the same pipeline. We isolated RNA, we constructed the libraries, and then we sequence with Illumina. And then we confirmed the expression of most of these genes. Of course, not all of them were indeed expressed on the capsaicin diet, which is okay, good. We confirmed the expression. And then we wanted to integrate our data sets because we know that bulk signals are coming from a heterogeneity sample, which is formed of many different cell types. So we perform first deconvolution. That means breaking down all the bulk sample into different cell types, as we do for the single nuclear RNA-seq analysis. And for that, we apply two deep learning approaches. One is tape in order to deconvolve. So we basically take our single nuclei as a template to collect the gene markers that we use for annotation, and then we compare these expressions in our bulk sample. And then we use a variational autoencoder model to generate synthetic single cell profiles using the bulk expression profiles, as well as our estimated cell proportion from tape and our collapsed data set from the single cell or single nuclei sequences. So we create this data set, a comprehensive data set, where we can see there is a clear correlation between the deconvoluted bulk and the single nuclei of reference. That means our brain collection was good. And then we see the proportions of cells, it's similar among all our cell types. And then we constructed our synthetic single cell RNA-seq that we can use to confirm not only the expression pattern, but also the gene expression pattern in a particular cell type. So now, we understood some transcriptional signatures, the escape, the clock, but then what is triggering this behavior? So for that, we use knockout of chemosensory system. Since we know that the gut is not driving this behavior, nor the metabolic needs, our hypothesis is that the chemosensory system is driving this behavior. So we wanted to knock out the olfactory system and the gustatory system. So we define olfaction and taste as the recognition of components in the environment that are either volatile or non-volatile compounds. The case of olfactory is those are that can be volatile at the room temperature. And we use KIP 2.1, which is an in-wire rectifier channel, in order to desensitize the neurons all the time through calcium influx that will cause differential negativity in the neurons that will basically block their firing potential. So what I want to say here in brief is that we're using this channel under the expression of these cell types in order to knock out the sensory systems. So I'm happy to tell you, because I was really shocked when we did the experiments, that DR64F neurons, the silencing of these neurons basically abolish this trait, indicating that they are related to the Capsaicin recognition, which is also present when we analyze the binary decisions. So we can say that the silencing of DR64F neurons abolish Capsaicin taste and preference. So now we now know that gustatory neurons are related to Capsaicin sensation, particularly this subset of gustatory neurons called DR64F. But then what is triggering the neuronal activation? And I can't say we have a receptor yet, though we're working on it. But I want to recall one proposal that was actually done 15 years ago, because there is a possibility that the TRP channels can be expressed in gustatory neurons, and therefore acting by expressing these neurons and driving the Capsaicin behavior in flies in particular. So now we have resources, particularly genomic resources. So we visited the fly cell atlas, particularly for the proboscis, and here we can see sensory neurons, and these are just an annotated cluster, where we see that there is no overlap of expression between TRPs and nociception and those GR64F neurons, nor GR5A neurons, indicating that nociceptive related TRPs are not expressed in GR64F neurons. So we can rule out the possibility that the activation of GR64F can be driven by TRP channels. Okay, so that's pretty much. So what is the conclusion of my research? So we know that capsaicin alters feeding and activity in flies through a TRPV1-Independent mechanism that is not mediated by metabolic needs. We know that DSK and clock are major signatures of capsaicin desensitization, and the learning memory system is rewired differently for females and males. This was revealed by a comprehensive brain transcriptomic dataset. And then we also know that the chemosensory system through GR64F gustatory neurons is involved in capsaicin feeding behavior. So now, what are our future directions? So first, we want to understand what are the implicated receptors and the time it takes to desensitize them through receptor knockouts. We also want to understand the connection between the inhibition of feeding and the inhibition of activity. We want to understand also the connectomics, and this is something that is really important because we see that the brains are wired differently. And in a side experiment, we carry out an oviposition experiment where we found that the female flies have an opposite behavior for egg laying, where they basically are aversive to lay eggs on capsaicin diet. And this is nothing novel because there are some other neurons related to be activated and basically have a modulatory switching of their behaviors. So of course we need to understand more about this matter. And then lastly, we are resolving this and all this occurs, but what might be the evolutionary advantage of recognizing capsaicin? That is something that we need to ask ourselves.